I decided to make a supplemental video about ATCase since there was some very confused faces at the end of class on Thursday. So if we remember, ATCase is aspartate transcarbamylase, and ACTase is the first committed step for the synthesis of pyrimidines. And as you'll recall from introductory biology or perhaps from molecular biology, that the pyrimidines are the DNA bases C and T. Now, C and T, when they're in a single strand of DNA, will base pair with a complementary DNA bases to form double-stranded DNA, which we'll talk more about in the nucleic acid section coming up soon. Now, if you'll remember, C is complementary to G, and T is complementary to A. G and A are known as the purines. So why are we talking about this? Well, it's because, again, ATCase synthesizes the first committed step to pyrimidine synthesis. And so it turns out that both the downstream products of ATCase, specifically CTP, and one of the complementary bases to the downstream products of pyrimidines, purines, specifically ATP, possess the ability to modulate ATCase activity. So if we look at a Michaelis-Menten plot for ATCase, so if we look at the velocity as a function of the substrate concentration, what you'll notice is that the substrate saturation curve is linear instead of hyperbolic, which is indicative of cooperativity or allosteric regulation. And this is because ATCase has 6 catalytic subunits and 6 regulatory subunits. And so those downstream products, specifically C, and the complement, specifically A, have the ability to modulate the activity of the catalytic substrate by binding subunit by binding to the regulatory subunit. So again, when we have ATCase on its own, we see this curve. However, if there's an excessive amount of CTP hanging out in the cell, which indicates that there is an excess of pyrimidines in the cell, we don't want ATCase to make more pyrimidines because we already have so many that there's an excess, which is implying that there are more than enough pyrimidines in the cell to base pair with the corresponding purine. So that ratio of pyrimidines to purines is getting out of whack. So in this case, CTP is going to allosterically regulate ATCase by binding to the regulatory subunit, which would change the shape of the catalytic subunit so that the activity of the enzyme is downgraded or downregulated. So how does this, and we remember from lecture that this works by increasing the Km for the substrate. So the new curve for ATCase and the presence of CTP, or an excess of CTP, is to shift that curve to the right. Now, so let's say we have a high amount of purines available. Specifically, if purines of G and A are around in approximately the same ratio, then we can use A as a proxy for the concentration of total purines hanging out in solution in the cell. So then, if we have a lot of G and A hanging around, that means we're not making enough C and T to base pair with G and A. So for that reason, we would want ATCase to increase its activity and make more pyrimidines to base pair with the purines. And so if a cell does this, or ATP does this, by going and binding to the regulatory subunit, which changes its shape and therefore changes the shape of the catalytic subunits in such a way that the Km is decreased. Now, you may be wondering how the activity of an enzyme can be changed if we're changing the Km, not the Vmax. To illustrate this, let's take a look at one spot on the substrate concentration. So we'll say it's right there in that box. If we were to look at the velocity of the enzyme at the corresponding spot and the ATPase on its own, or in the presence of CTP and ATP, we'll notice that at this particular concentration, the activity of ATCase when bound to CTP, the velocity is lower than without CTP, whereas if we look at the curve for ATP, the velocity is higher than in the presence of ATCase. So so assuming that the substrate concentration in the cell is going to stay relatively constant, because the processes within the cell work to maintain a relatively stable concentration of substrates in the cell, then depending on the Km of the enzyme, the velocity that we see could change. So at a particular concentration, so let's say that this is the cellular concentration of substrates, shifting the Km to the right in the presence of CTP causes the velocity to be lower at that concentration and ultimately lower for a broader range of substrates, whereas the addition of ATP causes the velocity at that particular given cellular concentration to be higher, and it also causes you to see the velocity to be lower at a smaller range of concentrations than in the presence of ATCase. I hope that this explanation helps.